name's Nick Downing. I'm a second-year CMCB student in Gabe Zentner's lab. We're located in Myers Hall on the second floor, just up the stairwell. I thought I would show you around our lab just so you could have an idea of what our everyday workplace is like and talk about some of the ideas that we like to focus on. Very broadly, our lab is interested in transcription regulation. Specifically, we want to evaluate the different functions of the mediator complex. Now, mediator is a huge macromolecular complex made up of about 30 different subunits, and it is conserved in yeast all the way to human cells. Now, in a very concise statement, it serves as a functional bridge to relay signals from DNA-bound transcription factors to RNA polymerase II to allow for transcription initiation and regulated gene expression. So this is where the magic happens. This is our lab in all of its modesty. I'd like to show you each of these aisles individually. Here in the back, we do have our supply aisle. It has all the kits and reagents and also just plastic supplies that we would ever need for tissue culture. In here we have Gabe's bench with some PCR machines. As you can see, very clean, well organized. Good job, Gabe. Now if we come over one spot, we have the benches of two of our fellow GCDB students, or the Genome Cell and Developmental Biology program. So on this left side we have Mustafa Saleh, who is not here because it's late. We also have Bob Polycostra's bench, who is kind of our coding guru. And apologies for not including some of my other lab mates in this video. I'm making it kind of after hours and no one's here, so I am kind of the Nosferatu of lab. And here we have JT's bench, who is our lab technician and manager. And he actually asked me if he should clean his bench before I make this video, but I told him not to worry about it, to be as realistic as possible. Now I will say, this is not a messy bench. This is a very realistic bench, okay? Now we could contrast this with my bench, which I've shown here. But I have been warned by other graduate students that if your bench stays too clean, then your PI might start thinking that you're not doing any experiments and they might wonder why they took you in. It might not be a bad thing to just kind of move things around, kind of make a little bit of a mess, just so people know that you are still alive. So. You should be doing experiments though, because your PI does know whether or not you're actually putting in the hours. If you check out our lab on IU's website, or even at our lab-specific website, you will notice that much of our work uses budding yeast as a model system, or the Saccharomyces cerevisiae. Now these are a really cool model to work with. They're cheap, easy to grow, you can easily manipulate them, and if you feel like taking a break from lab work, you can actually just throw them in the fridge for a while. Now another model that our lab is beginning to use or dabble in is this Dictyostelium or slime mold. So this is related to the slime molds that grow on mulch, as you can see here. But since you're applying to a cancer program, you're probably wondering whether or not we work with cancer cells. Yes, we do. That is actually where Sung Yoon and I come into play. So Sung Yoon is the postdoc in our lab. Now we believe that there are specific mediator subunits that interact with oncogenic transcription factors that can then mediate kind of oncogenic or cancerous phenotypes. Sung Yoon and I are working together to evaluate disruption of these protein-protein interactions in order to see how that kind of curbs cellular phenotypes, morphologies, etc. Now, if these ideas hold true, the logic is that we could help develop inhibitors that could be used to benefit patients that come from these specific molecular backgrounds. We work with these cells in our tissue culture room, which is actually just about 5 feet from the front door of our lab. We do share this room with the Walzak lab. Let's go see it. Now, as I was saying, we do share this room with the Walzak lab, so they are also another cell biology lab in our department. So on one side of the room we have their hood, and on our side we have our hood. So again, this is just a typical hood, nothing that fancy about it, but it's designed to push everything out of the hood so that your skin cells, all the contaminants, do not go onto your cells. Okay, that is enough of that. And I'd like to give a little plug to Gabe's band called Tau Zero, now available on iTunes. So, if you are into metal, you should definitely check them out, and I can assure you that this is in fact Gabe. Well, that is it for me. Thank you guys for watching. Congratulations on being interviewed. And remember, you got interviewed because we like you. So please don't feel any pressure, but always stay sharp. Thanks.